Dear distinguished audience, thank you for taking your time and visiting our webinar, in the first part of which I will talk about how to transfer the electrophysiological kind of recording in the nervous tissue to the domain of imaging by way of multiphoton microscopy, combining the advantages of the two techniques, namely high temporal resolution with detailed three-dimensional spatial information. Traditionally, two-photon microscopy, the basic physical principle of which you may see on the upper left panel, showing how the high-resolution point spread function comes about, allows for detailed physiological recordings, as you can see on the videos, through the recording of calcium signals detected by calcium indicators like GCAMP that can be seen on the lower left panel when bound and unbound to calcium. The recordings are spatially detailed. You do see the cell bodies lining up, but also thin processes like dendrites down to the level of dendritic spines that have a diameter of roughly 1 micron. These recordings were done in the visual cortex of awake mice during behavior. At the same time, we do know that the activity that we do see here, calcium activity, has sort of a murky relationship with the electrical activities of the cells that traditionally are regarded as the per se activity of the cells. As an in vitro example, here you see the intracellularly recorded traces of 5 evoked action potentials, and here their calcium signal counterpart. The relationship is apparent, but there is some lag, and the temporal resolution is far from revealing the underlying individual spikes. Scientists, we among them, have tried to establish a firm relationship between the two signals, taking into account the characteristics of the indicators as well. They constructed complex algorithms to try and decipher spike trains behind the calcium signal, using pages of complex equations like the ones you can see here. They came up with solutions like that on the lower right panel, where the spike trains of hundreds of imaged cells are reconstructed from the recorded calcium signal. This looks great and pretty, but it is still possible that the two kinds of signal may bear somewhat different information too, which, in that case, may be lost in translation. Nowadays, there is a new advent after some evolution, as it is proven by the lower left paper, of different indicators. Ones that bound to the membrane and report potential differences, and especially the change of it. After years of struggling, now we can safely say that the newest indicators are very well usable, even in vivo, awake, behaving experimental settings, and they are available as genetically encoded indicators. Currently, we are using mainly two of these: the JEDI family with the JEDI 2P in the upper left corner, and the ASAP family, upper right, especially ASAP 5. But obviously the field actually changes literally by the hour. Classical multiphoton, two-photon scanning techniques are the galvo-galvo and the galvo-resonant scanning. They are not particularly slow, since they can record physiological data. Their speed ranges from a typical 20 through 40 to a few hundred hertz, can reach 1,000 hertz if recording a smaller area. As we demonstrate on these traces, these speeds are not enough to record the details of the voltage signal. Small wonder, they were designed to deal with calcium signals. But here we have the new acousto-optical scanner-based three-dimensional Femtonics 3D AO scanning microscope. And look, we may achieve the speed that is required to record those voltage signals. What we do achieve this speed with which is required by the nature of the voltage signal, is a special kind of AO scanning technique called the drift scan. You see it here in the upper corners, but I also talk about them in details in an earlier Atlas webinar that you can find at our webpage. There are other pretty informative webinars there too. Do check them out. The bottom line here is that during the 30 or 10 microsecond AO cycle, we can record lines as long as 300 micrometer. So we may achieve 30 to 100 kilohertz scanning rate, even on multiple cell bodies or other cell parts situated in 3D. This speed also allows for a crucial feature when it comes to high-speed recording, voltage recording included. In the second half of this webinar, we will detail this feature, the real-time motion correction, which you can see on the lower right corner. This all is achieved by the Femto 3D AO microscope that you can see in the middle. Note the ample space below the objective, allowing to place complex behavioral setups under the microscope. This recording was done with such a setup: awake, behaving voltage sensor recording. On the left panel, you may observe the expression. The membrane is depicted nicely by the sensor. Choosing one of these cells, the trace, even with a recording speed around 1 kHz, looks like a genuine electrophysiological recording with high amplitude action potentials. The average trace of APs are on the right panel, with all distinguishable, even subthreshold parts. This average is taken from a recording which had a higher temporal resolution, 3 kHz. Here it is in the lower right corner. This graph here shows in a very plastic manner how the calcium signal that we have recorded so far as physiological signal relates to the membrane originated voltage signal. Here it is. By recording more than one identifiable cell, like in the former recording, in a genuinely three-dimensional arrangement, one cannot help saying that we are in fact recording optical electrophysiology, therefore opening a whole new avenue for neuroscientific and translational research. Having said all that, now I give the word to my excellent colleague Katalin Ócsai, and with that, I myself thank you for your kind attention.
Welcome everyone. Thank you for taking your time to join this webinar about Femtonics FocusPinner, a solution for 3D real-time motion correction for in vivo measurements. I'm Katalin Ócsai, the head of software and AI department in Femtonics Ltd. And the developer of real-time motion correction. As it was mentioned in the lecture about voltage-sensitive dyes, the main advantage of the acousto-optical technology is the flexibility that the position of the focal point can be controlled within a 10 to 30 microsecond temporal precision without any mechanical constraint. The technical background is that we have acousto-optical deflectors, which consist of crystals bonded to an electric transducer emitting ultrasound waves to the crystal when driven by a sine wave. Changing the frequency of the sinusoidal voltage changes the deflection angle of the light falling through the aperture. With this technology, a very fast random 3D access can be achieved, making it possible to scan arbitrary ROIs. As a consequence, with acousto-optical technology, we can scan very complex, almost arbitrary 3D ROIs following the shape of the objects to be measured with a very high temporal resolution. In example, we can achieve 100 kHz in case of point scan and 16 kHz on a 50 micrometer long ribbon. So we can say that one of the greatest advantages of the acousto-optical technology is the incredible temporal and spatial flexibility, which is actually a crucial feature for the real-time motion correction. But why is real-time motion correction essential in vivo measurements? Well, the brain movement due to intended and visceral motion, such as heartbeat or respiratory movement, can cause very huge artifact in the calcium signal, as shown on the right, especially when the ROI is relatively tight and the scanned object can move out from the scanned area or volume. So what is the main concept behind the Femtonics FocusPinner? We select a reference point, which can be either a fluorescent bead or even a bright cell, and we measure 3 orthogonal AO drifts on the reference point in X, Y, and Z directions and calculate the intensity profiles of the drifts, then determine the center rates of the profiles. Reference and imaging data are collected sequentially according to the ratio, which can be set by the user. When the animal moves vividly, it is worth having a larger fraction, but generally 10% is a good choice. Then in the FPGA level, the firmware calculates the dislocation vectors between the center rates of the intensity profiles of subsequent reference drifts in each direction, X, Y, and Z. Based on this estimation, the movement will be compensated on the next imaging drifts by shifting the target of the focal point. Since a very fast, less than 1 millisecond intervention is needed, calculations are performed not on the personal computer level, but on the electronic cards controlling photomultiplier and acousto-optical crystals. Using this idea, the Femtonics FocusPinner can compensate the motion of the cell population quite robustly. Moreover, keeping the neuron in the ROI does not need to increase the size of the squared ROIs, which would mean a significant reduction in the sampling rate. Actually, the frame rates decrease only with the user predefined fraction between reference and imaging drifts. Z-stacks are quite often used for structural imaging of a dendritic tree. To avoid blur layers, just enable Femtonics FocusPinner. It will compensate the movement during scanning each rows of the layers and movement between scanning the layers as well. Real-time motion correction can be crucial in case of dendritic measurements as well, where a narrow ribbon is selected as an ROI. This movie shows a recorded ribbon scan as long as 20 minutes, which was used to elicit signal from dendritic splines. The compensated motion along X, Y, and Z axis can be stored in the measurement file in 3 curves, so the corrected movement can be analyzed without finding any artifact of the signal extracted from somata or dendrites labeled with calcium or voltage-sensitive dyes. Here we can see the stored movement curves. The x-directional movement was plotted with yellow, the y with orange, and the z with red. At the bottom of the page, calcium curves elicited from a dendritic spine can be seen. The green one shows the signal from measurement with real-time motion correction. Then the stored motion was applied on the dendritic measurement, and signal elicited from a moving spine is plotted with blue. We can see that without motion correction, the blue signal missed some of the active periods and suffers from huge artifacts caused by motion. Finally, let's see some quantitative analysis. Without the FocusPinner, calcium signals are quite noisy in the scanned population. Moreover, it's very hard to distinguish between the movement artifact and action potentials. However, when enabling real-time motion correction, artifacts disappear and transients with regular rise and fall appear as calculated and modeled from the measured data. Quantitatively, a large-scale improvement can be seen in the signal-to-noise ratio. As a summary, let's see the main advantages of Femtonics FocusPinner. It is acousto-optic based, so by leveraging the capabilities of 3D random access line scanning, our system enables seamless motion correction along each axis. It offers a significant advantage over the currently available XY plane cross-correlation based solutions by providing a feasible Z-directional correction. It is useful at in vivo measurements. Motion artifacts caused by the movement of the animal, such as respiratory movement or heartbeat, can be efficiently eliminated with an average residual motion under 10% of the original motion. It is real-time. During the measurement, the focal point is online relocated based on the estimated motion of the reference point. Compared to quasi-real-time solutions, the reaction time is 1/10. The cycle of the repeated motion correction is less than 100 microseconds, meaning that the largest residual motion amplitude is the one collected during this short time window. It is compatible with all scanning modes, in example, point scan, raster scan, chessboard scan, multi-cube scan, longitudinal and transverse ribbon scan, snake scan, and Z-stack. So, the all scanning modes of the Femto 3D Atlas system. Thank you for attending this webinar. I hope you enjoyed it. If you are interested in more details or in an online demo, please write an email to the following address. Goodbye.